In this video, we're going to learn about the cell cycle. What is the cell cycle? It is a series of repeated steps that each cell takes every time it doubles. So I've kind of tried to represent a cell, and here's a cell, that's the nucleus. It goes through these cycles, this cycle, and then at the end of the cycle, it divides into two cells. Now, a cell, particular cell, can divide up to maybe an average of 50 times, after which it won't divide anymore and it dies. And the reasons for that are beyond the scope of this course. But suffice it to say that every time the cell divides, it has to go through exactly the same steps. That is called the cell cycle. Now, the cell cycle has stages in it. The first stage is called the gap 1 phase, and it's represented by G1. So G is the acronym used to indicate gap. This is where things start. So when a cell enters the cell cycle, first it has to go through the G1 phase, where the size of the cell increases and the cell is producing all the different components that it requires to get ready to go through the next stage. The next stage, which is the S phase, stands for synthesis. That is the stage in which the cell doubles its genetic material. So the cell copies each and every chromosome, and if you remember from a previous video, generates sister chromatids that are attached through the centromere. After the DNA synthesis is done, another preparatory and growth phase happens, which is called the gap 2 phase. And the cell going through preparation steps until it reaches the mitotic phase. Now, starting from the G1 phase all the way up through the mitotic phase, there is absolutely no cell division steps happening. This is all preparation. And just like any event that is set up to be successful, there is a lot of preparation involved. So all these preparatory steps all together are termed the interphase. That's because it's the step that comes in between the actual division steps. Okay, so we got the G1 phase, the S phase, G2 phase. Then we go to the mitotic phase. And the mitotic phase has two stages within itself. First is mitosis. Mitosis is a stage at which the cell divides the duplicated genetic material into half and send them to the two opposite ends of the cell. So mitosis is nuclear division. Then cytokinesis comes right after mitosis, in which actually the cell cytoplasm divides and gives you two cells. So all the way through, starting from the G1 all the way to the end of mitosis, we're all looking at everything happening in one cell. And it is only upon cytokinesis that you end up having two cells that are identical to each other and to the cell that it started. Now let's take a look at what the chromosomes would look like. So I'm going to go to my pen. So in here we have, if we were going to draw the chromosome, I'm going to try my best to draw a chromosome with this pen, would be like this. It would be the arms and the centromere. So the chromosome is going to look like this all the way throughout this stage, but by the time, so even at the beginning of the S phase, it looks like that. It doesn't have any sister chromatids. But when it goes through the DNA synthesis, the S phase, when it comes out, it'll look like this. It will have a sister chromatid that is attached to it by the centromere. So by the time the cell finishes the S phase, its genetic material has doubled. And then These cell goes through the G2 phase, and during mitosis, what happens, these two sister chromatids are separated from one another. So by the time mitosis is finished, one sister chromatid goes to one cell. Oops, sorry. My centromere is a little ugly looking. And then another sister chromatid goes to the other cell. So we started here with a cell with unreplicated chromosome. It goes through the S phase, it doubles its genetic material. Then by the time end of mitosis happens, we got two chromosomes on the two ends of one cell, and then upon cytokinesis, you get two cells that have the exact genetic material and the exact number of chromosomes as what you started with. So here's a question for you. Prior to mitosis and after S phase, each chromosome of a eukaryotic cell consists of a pair of identical structures called, what would you say the answer to this be? Draw a pair of homologous chromosomes before they enter into S phase and label the different parts. Now draw the same chromosomes after they complete the S phase. So you're only using one pair of homologous chromosomes. Then draw it for two pairs of homologous chromosomes, and then draw it before and after the S phase. But it is important that you sit down and do this problem.